有啲同學咧就寫咗字又當答咗嘅，呢個答法都有少少躺平，有啲軟對抗嘅感覺喎。
做這種事有咩好處
？2022年第9條題目係有關於抗生素啦、分子遺傳學啦同埋生物科技嘅。咁題目就講啦，抗生素咧係廣泛使用於治療細菌感染嘅，而抗生素啦係可以殺死細菌啦，又或者啦係抑制佢嘅成長啦。然而啦，有啲細菌菌株咧就對抗生素咧出現咗抗性。咁 Part A 咧都問翻啲好直白嘅題目啦，亦都好基礎嘅，就係問翻啦。抗生素係點樣殺死細菌同埋抑制佢哋嘅成長呢？就寫出3個可能嘅方法啦。嗱，呢條題目咧都係嗰句啦，就考你有冇睇清楚題目嘅啫。因為真係有同學咧唔知做乜鬼揞眼，人哋就問你抗生素，佢就答咗抗體嘅。咁當然啦，又或者啦，你真係睇得到咧，佢係問你抗生素嘅，不過你寫嘅功能咧就寫咗抗體嘅功能喎。咁啊，快手睇翻片又溫下書啦，好短嘅啫，1分鐘咧就幫你分清楚2款嘢噶啦。咁而家啦，真係睇清楚啦，題目係問抗生素喎。咁抗生素有咩嘅功能呢？咁書本啦都好直接了當俾晒你啦，題目亦都一樣。不過有一樣嘢我想提大家嘅，去點樣抑制細菌去形成細胞壁啊，破壞細菌嘅細胞膜啊，抑制細菌去做一個蛋白質嘅合成啊，又或者抑制細菌呢佢嘅核酸嘅合成啊，呢個都只不過係故事嘅頭半段嚟嘅啫。因為題目係講到明啦，係殺死細菌同埋抑制佢嘅成長，所以啦，唔該大家咧都分清少少咯。邊個係殺死佢呢？邊個係抑制佢嘅成長呢？喺個答案嗰處咧，你要留意翻嘅就係藍色嘅部分啦。嗱，呢啲嘢咧有陣時書都冇寫得咁清楚噶，因為咧書都淨係講翻啦，殺死咯，同埋抑制細菌成長咯，就講咗個原因咯。但係個真正嘅結果啦，究竟係殺死佢啊，定係抑制成長呢？原來啦，抑制佢嘅細胞壁形成咧，係抑制佢嘅成長啦。破壞佢嘅細胞膜嘅話咧，就係導致到個細胞係會分解咗啦。抑制隻細菌去製造蛋白質嘅話咧，就係抑制咗佢嘅成長啦。抑制嘅細菌佢嘅核酸合成咧，其實就係令到佢哋做唔到繁殖啦。即使佢入咗你嘅身體入面啦，都唔怕佢1變2，2變4去整到你病咯。咁有關於抗生素啦，有啲咩題型變奏呢？第一款啦，自不然問翻啦。當我哋去發現到抗生素呢一款東西嘅時候啦，究竟我哋經過咗啲乜嘢嘅科學探究嘅步驟呢？係嘛？咁你可以睇翻呢一段片啦，我都會擺翻喺留言區咧，俾你哋做參考嘅。第二個問題啦，就係睇翻上面呢幅圖咯。當阿費林明當初啦，佢所謂發現咗呢個抗生素啦，咁其實佢點解咁肯定啦？呢一樣物質就唔係啲真菌佢哋自己，而係有一款嘢由真菌去釋放出嚟嘅呢。而呢款嘢係可以去散播開，從而停止咗、抑制咗細菌嘅成長呢。咁其實睇翻呢幅圖呢，你大約都知道個概念噶啦。如果隻真菌喺呢樹，啲細菌喺呢樹，如果而家我哋講緊所謂嘅抗生素，其實就係隻真菌自己嘅話呢，咁即係話要隻細菌同嗰隻真菌有一個正面衝突，行埋一齊啦，佢哋先會打交嘅啫。但係而家我哋喺呢幅圖，我哋發現啦，喺個真菌嘅附近呢，其實都係冇細菌嘅。咁即係話啦，應該係有一樣嘢就係隻真菌釋放咗出嚟，而呢一樣嘢係可以散播開去，就形成咗好似一個保護罩咁嘅款啦。咁所以啦，喺個真菌周圍嘅部分呢，就相對上就會少一啲細菌嘅生長咯。跟住啦，就去到 Part B 啦。細菌 R 咧就係具有抗生素抗性基因嘅致病病菌嚟嘅。咁佢嘅基因產物咧就可以將抗生素 X 咧去分解啦。而而家咧啲科學家咧就提出一個嶄新嘅方法去對付細菌 R 咯。呢個方法咧就係利用咗合成嘅多核苷酸啦，佢就會同翻轉錄抗生素抗性基因嘅 mRNA 去結合，從而啦抑制咗呢個基因嘅表達嘅。 咁我哋啦就將呢個合成嘅多核苷酸同埋抗生素 X 一齊用嘅話呢，就可以殺死啲細菌咯。咁下面呢幅圖呢，就顯示翻啦呢個合成嘅多核苷酸嘅鹼基序列啦，呢個 mRNA 信使 RNA 嘅部分鹼基序列啦。咁啊第一部分就問翻咯，合成多核苷酸同埋呢個信使 RNA 嘅邊一啲嘅鹼基係會結合呢？咁我哋呢，就喺翻嗰個鹼基序列呢，畫翻條線咯。呢個題目啦就係考翻我哋有關於鹼基互補嘅概念啦。G 配 C，A 配 U，A 配 T 啦。咁喺個 mRNA 入面呢，就冇 T 嘅，得 U 嘅啫。咁我哋就睇翻呢個合成嘅多核苷酸啦，同埋呢個信使 RNA 咯。合成嘅多核苷酸呢，佢就係 AGT、GAC、TC、GGTC 同埋 AGC 啦。咁我哋根據翻啦 G 配 C、A 配 U、A 配 T 嘅鹼基互補嘅概念呢，咁我哋就知得到啦。信使 RNA 呢，會係邊一 part 嘅鹼基序列俾人哋去結合咗啊？自不然就係UAC、CUG、AGC、CAG 同埋 UCG 啦。咁樣睇翻題目間翻低咧就搞掂噶咯。咁其實過往咧都有唔少題目咧係問翻我哋呢啲鹼基互補嘅概念嘅。咁啊快手睇下片溫下書咯。然後啦，去到第二部分咧，就我哋解釋翻呢個合成嘅多核苷酸係如何去抑制到抗生素抗性基因嘅表達咯。呢個題目咧就考翻我哋有關於轉譯嘅概念啦。轉譯有啲咩嘢係參與喺其中呢？信使 RNA 啦，轉移 RNA 啦，核糖體啦。咁當中啦有啲咩嘅核心概念我哋一定要識呢？轉譯就係喺呢個細胞質嘅核糖體嗰度發生嘅，而喺個核糖體就將嗰個信使 RNA 啦，同埋呢個轉移 RNA 啦，就拉埋一齊啦。但係轉移 RNA 同埋信使 RNA 點樣認到對方先？睇翻佢哋個密碼紙同埋反密碼紙嘅，其實都係一個鹼基互補嘅概念嚟嘅啫。咁而家題目就講啊嘛，我哋有一個合成嘅多核苷酸，佢就會黐鬼住咗呢個信使 RNA， 咁黐住咗咯，我哋就要指出得到轉譯就將會失敗啦。當呢個合成嘅多核苷酸，佢就會同我哋呢個 mRNA 做個結合。所以啦，呢個轉移 RNA 就唔能夠再黐埋去呢個信使 RNA 嗰處，咁亦都唔能夠傳送一個相對應嘅氨基酸啦。咁所以啦，氨基酸之間亦都唔能夠做一個結合，所以啦，呢隻細菌就不能夠生成呢個蛋白質，而呢個蛋白質正正就係我哋呢個基因產物啦，係咪啊？所以啦，轉譯咧就唔能夠喺呢個結合咗嘅位置咧去進行啦。跟住去到第三部分題目就叫我哋提出翻利用呢個合成嘅多核苷酸啦，去對抗抗生素抗性細菌嘅一種優勢，有啲咩嘅好處咯。咁題目要考我哋嘅呢，就係有關於正常嘅細菌，如果佢真係有抗生素抗性嘅話啦，我哋遇到咩問題呢？就係令到我哋原本用開嗰隻抗生素未變得冇效啦，係咪？但係而家唔同啦，哪怕佢嘅基因有突變咗啦，可能咧令到佢嘅抗藥性咧強咗或者弱咗都好，唔緊要，變咗我都唔怕，因為只要我知道咗呢個抗藥性嘅基因，佢嗰個鹼基序列嘅時候咧，我就可以隨時設計到一條新嘅合成多核苷酸，都係黐翻落去呢個信使 RNA 個樹，我都係能夠停止咗呢隻細菌進行呢個轉譯嘅作用。同意啦，佢亦都唔能夠產生到呢個基因產物出嚟啦。第二呢，又係考翻我哋分子遺傳學嘅概念啦。合成嘅多核苷酸點解佢咁有專一性呢？係因為佢有鹼基互補嘅概念啊嘛。一定係 A 配 U、G 配 C、T 配 A、C 配 G 噶啦。設計咗呢個合成嘅多核苷酸嘅時候，一嘢黐落去，我哋就能夠抑制到嗰個目標基因嘅轉錄同埋轉譯咯，係咪啊？有關呢條題目呢，有啲常犯錯誤都想提一提大家嘅，咁啊自不然又要提大家啦，直線抽擊答題法啦。有啲同學呢，就寫咗字又當答咗嘅，例如啦，今次人哋題目就講啦，喂，利用呢個合成嘅多核苷酸去對抗呢個具有抗生素抗性嘅細菌嘅優勢，佢就寫啦，有效咯。佢唔會有不良嘅效果噶，又或者啦，佢對個人咧係冇害噶。題目都講到明係優勢啦，咁梗係有效啦。但係問題你要講係點樣有效法啊嘛？或者啦，佢咧對人體冇害嘅， 個題目又唔係要你做比較，我要你比較翻呢，去食抗生素同埋啦，去用呢個合成嘅多核苷酸有啲咩嘅分別，又冇問過。又或者有啲同學呢，就直接呢就抄題目嘅啫。佢呢就能夠對抗到呢個具有抗生素抗性嘅細菌啦。人哋問你就係佢點樣去對抗佢啊嘛，有咩好處啊嘛？你話係咯，因為對抗到咯。呢個答法都有少少躺平，有啲軟對抗嘅感覺喎。咁當然啦，有啲同學就直頭就係冇睇到題目啦，又係答翻有關於我哋嘅免疫系統啊，我哋嘅抗體嘅概念啦。因為佢哋由 Part A 已經睇錯咗抗生素變咗做抗體啦嘛，又或者佢搞唔清抗體同抗生素嘅分別啦嘛。咁所以睇題目小心啲咯。好，咁啊又嚟到一點出發啦。今次嘅題目呢，就由細菌感染開始嘅，考我哋兩樣嘢。第一就抗生素啦，咁啊先講抗生素啦。今次啦都講到明係有呢個抗藥性嘅細菌株嘅。咁原因何在呢？其中一個原因啦就係濫用抗生素啦。而當中啦點解要講埋選擇壓力啊？其實呢個都係一個人工選擇嚟噶嘛。係因為而家我哋係濫用抗生素，所以佢係一個人工選擇嚟嘅。咁所以我哋就可以講選擇壓力咯。其實個選擇壓力咧就係講緊嗰個抗生素嘅劑量啊。而嗰啲細菌呢，佢經過基因嘅突變啊，從而啦就令到佢嘅抗藥性，有啲細菌可能勁咗，有啲細菌可能渣咗嗰啲，咪會俾個抗生素殺咗佢咯。強一啲嘅抗性嘅就自不然能夠頂得順呢個抗生素。而整個故事就係點啊？佢可以生存，可以繁衍，將呢個抗藥性呢個特性呢個好處傳俾下一代。就令到每一代就可能再有突變，就再強啲，直至到一刻嗰隻抗生素完全對佢冇效啦。同一時間咧就梗係講埋抗體啦，因為有啲同學就係兩隻字分唔清啊嘛，係咪？咁抗體同埋抗生素有啲咩比較，大家都要溫一溫書咯。咁過往啦，除咗抗生素咧，都有問抗體嘅題目嘅，大家快啲睇下片啦噃。然後啦就去到分子遺傳學啦，自不然考我哋嘅就係轉錄轉譯啦，係咪？大把題目由12年去到而家22年都有噶，咁啊亦都有呢個蛋白質合成啦，係咪？都仲未計長題目，我齋俾你睇嘅都只不過係 MC 啫。然後啦兩者結合咧就有關於生物科技啦。其實利用真菌佢釋放出嚟嘅抗生素去對付細菌呢，已經係一款好典型嘅生物科技啦，係咪啊？而除此之外呢，我想再講多些少嘅就係有關於基因重組技術啦。當中亦都有轉基因生物啦。今次我想俾大家嘅例子呢，就係可食用嘅疫苗啦。今次我哋利用生物啊嘛，利用咩啊？利用香蕉樹。咁我哋呢，就先係能夠揾得到一隻病毒，佢外面呢，就有唔同類型嘅蛋白質嘅。呢啲蛋白質呢，都係由個病毒去整出嚟噶嘛。咁例如啦，呢個 GP5。呢個 E protein 嘅個病毒入面啦，就當然有一啲遺傳嘅物質係能夠製造到呢個 GP5 嘅 E protein 啦，係咪？咁我哋啦就將呢個基因咧抽取出嚟，就同個基質去做一個合成基因重組技術，然後放落去一隻細菌個樹，再感染呢啲香蕉嘅細胞。咁啊，將呢個香蕉細胞就改變咗佢，咁改變咗嘅呢個香蕉呢，佢就係轉基因生物啦，係咪？咁佢有咩咁特別啦？佢就係能夠有呢個 GP5 嘅基因，就能夠製造到呢個 GP5 嘅 E protein，從而令到呢棵香蕉樹佢所產生嘅香蕉啦，你未必會食香蕉葉嘅，係咪？喺呢個香蕉啦，佢就有呢個 GP5 嘅基因，亦都能夠產生到呢個 E protein。所以啦，當我哋食呢個香蕉嘅時候呢，就好似食咗呢個 E protein 落肚啦。但係嗰隻病毒就會入去你身體啊？冇啊嘛。咁所以啦，你就純粹呢就獲得呢個 E protein，但係佢又唔係你嘅嘢，係咪會引起呢個免疫反應啊？所以呢個呢，就係可食用疫苗嘅機制咯噃。而有關於生物科技啦，自不然係要講下道德議題啦，或者一啲潛在嘅危機啦。又講生化危機呢隻遊戲。其實不是夢嘅，佢總有一日係會發生嘅，所以有咁多人係關注生物科技佢嘅安全問題啦。同埋有關道德議題咩事呢？今次都係轉移嘅意思。今次我哋轉移嘅係香蕉樹啫，但係如果下次我轉移嘅係一條魚啦，係一個番茄啦，係一隻貓啦，一隻狗啦，甚至乎我哋轉移嘅係人嘅話，咁究竟啲人會點睇呢？ 2022 question 9 is a question about the antibiotics molecular genetics and biotechnology. Antibiotics have been widely used to treat bacterial infection. They work by killing bacteria or inhibiting their growth. However, some strains of the bacteria have developed resistance to antibiotics. So for part A, a very straightforward and a very fundamental question. How can antibiotics kill bacteria or inhibit their growth? State 3 possible ways. So in this question, it checks us, do we read the question carefully? We need to distinguish antibiotics from the antibodies because some students, they really misread the question or they cannot distinguish antibiotics from antibodies. So that's why I watch the video or do the revision for the antibiotics and for antibodies. So after we really read the question very carefully and then we know that it's really asking antibiotics, and then we need to recall the function of the antibiotics. So you can see that directly copied from the book. Inhibit the formation of the bacterial cell wall, damage the cell membranes, inhibit the synthesis of nucleic acid or the DNA, inhibit the protein synthesis in the bacteria. However, there's something I would like to add. How do these methods kill the bacteria or inhibit their growth? So you need to do the matching. Of preventing the formation of the bacterial cell wall, it can inhibit the growth of the bacteria. Damage the cell membrane, cause lysis of the bacteria. Inhibit the protein synthesis, DNA synthesis, it can inhibit the bacterial growth or stop them from reproduction. Therefore, the bacteria cannot do the cell division 1 to 2, 2 to 4, 4 to 8, and then the population of the bacteria will not be that large and cause any disease in your body. After we talk about the antibiotics function, so any possible question variation, surely we can ask you the discovery of the antibiotics, any scientific investigation, the steps you can recall. Observation, asking questions, doing experiments, something like this. So you can watch this video for revision. And then we can take a look at this diagram. And according to this diagram, why the framing suggests that it was not the fungus itself, but a substance spreading from the fungus, produced by the fungus, that stopped the bacteria from growing. So you can imagine the case. What if that particular chemicals or substance is the fungi themselves? Therefore, the bacteria, they need to grow, and then grow to the fungi, and then they have the direct contact, direct battle, right? However, we find that there is no bacterial growth around the fungi, surround the fungi, beyond the fungi. Therefore, there may be some substance produced or released by the fungi, and this chemical spread from the fungi and stop the bacteria from growing. And then for part B, Bacterium R is a pathogenic bacterium which possesses an antibiotics-resistant gene, and its gene product can break down the antibiotics X. And scientists have suggested a new approach to fight against Bacterium R. This new approach involves the use of the synthetic polynucleotide, which binds to the mRNA transcript from the antibiotic-resistant gene, and the expression of the gene is then inhibited. By administering this synthetic polynucleotide together with antibiotics X, Bacterium R can be killed. You can see the base sequence of the synthetic polynucleotide and part of the mRNA below. So we have the base sequence AGT, GAC, TCG, GTC, and ACG. And then for part 1, on the mRNA, we need to underline the base sequence to which the synthetic polynucleotide will bind. This question, it checks us the concept about the complementary base pairings, GC pairing, AU pairing, AT pairing. And in mRNA, it has U instead of T. So you can take a look at the base sequence of the synthetic polynucleotide, and then we do the complementary base pairing. We have UCA, CUG, AGC, CAG, and UCG. And then we can underline the base sequence on the diagram. And there was question about the transcription, translation, which also checks us the concept of the complementary base pairings. So watch the video for the revision. And then for part 2, explain how the synthetic polynucleotide can inhibit the expression of the antibiotic-resistant gene. So we need to recall the mechanism of translation. What are the roles of the mRNA, tRNA, ribosome in protein synthesis? So the critical concept is that translation occurs in the cytoplasm where ribosomes are located, and the anticodon of the tRNA bind to the complementary codon of mRNA. So the ribosome, mRNA, tRNA, they work together for the protein synthesis. And now from the question, we know that the synthetic polynucleotide will bind to the mRNA. After binding, the single-strand template is no longer available, so the tRNA cannot bind to the template to transfer the corresponding amino acid. That's why the amino acid, they cannot bind together to form the protein. Therefore, translation cannot proceed at the binding region. So you need to recall the important role: mRNA, tRNA, and the translation, this concept. And then for part 3, suggest one advantage of using synthetic polynucleotide to fight against antibiotic-resistant bacteria. This question, it checks us, how can we realize the problem of the antibiotic-resistant bacteria? So the bacteria with higher antibiotic resistance make the antibiotic ineffective. But what is the reason to make this higher antibiotic resistance? It's about the gene, the mutation of the gene, and then the gene product can break down the antibiotics X. However, now we use this new method, the synthetic polynucleotide bind to the mRNA. Concept is that once we know the sequence of the antibiotic resistant gene, we can design a new synthetic polynucleotide, and it will bind to the mRNA, and then it will inhibit the translation, and then there will be no gene product produced by this bacteria. Therefore, we can still use the antibiotics X, and we can recall the concept of the specificity of the synthetic polynucleotide. The action is very specific as it targets the resistant gene, and this idea relies on the concept of the complementary base pairing. And I would like to mention some common mistake. Most of the students, they just give very vague answer, very ambiguous, such as it's effective, it has no adverse effect, and it is not harmful to the humans. So the question is not asking the comparison between using this new method and using the antibiotics. Therefore, you do not need to talk about it's not harmful to the humans. And then it is effective, it has no adverse effect. Actually, you are not answering the question because it asks about the advantage. Therefore, please watch the video about the straight to the point. And then some students simply repeat the question stating that it could fight against antibiotic resistance. So they are not answering the question as well. Or some students did not read the question carefully and answer it will not cause any immune response and that the body will have a higher level of antibodies, so which are related to the body defense. They may misread the question in part A already. For the curriculum mapping, the question starts from the bacterial infection, and then it asks about the antibiotics and the molecular genetics. And for the antibiotics, surely we can talk about the abuse of the antibiotics and need to the antibiotic-resistant bacteria to develop. And this process is a artificial selection, and the selection pressure is the presence of the antibiotics, the abuse. And then some bacteria, they have the mutation in the gene, they may have stronger or weaker antibiotic resistance ability. And then the bacteria with a higher antibiotic resistant ability, they can survive and reproduce and pass this favorable characteristic to next generation. And then the population of the bacteria with higher antibiotic resistance will increase. Finally, the antibiotics will become less effective and less effective. We call the idea of the abuse of antibiotics. And then surely we can compare the antibiotics and the antibodies. And then for the molecular genetics, for the transcription, translation, for the protein synthesis, you can see that a lot of MC in the past year. There were long questions about the molecular genetics as well. So it is a hot topic. And then we combine the idea, it's about the biotechnology. Discovery of the antibiotics and use the fungi to produce the antibiotics to fight against the bacteria is a very classical example of the biotechnology. And in this question, I would like to talk about something more is about the recombinant DNA technology, the transgenic organisms. And this time, I would like to introduce the edible vaccine. So in this diagram, you can see that the mechanism of the banana vaccine. So once you eat the banana, you intake the vaccine as well. Is the concept about the edible vaccine. So we have the virus here, and then we find that there are some protein coat on the surface of the virus. For example, GP5, the E protein, and there must be some genes coding for this GP5 E protein for the virus itself. Therefore, we extract the structural gene of the GP5, and then we use the recombinant DNA technology to put this gene to the plasmid. And then we put this plasmid to the bacteria to transform it, and then we use the bacteria to infect the banana cells. The bacteria and for the banana, they are the transgenic organisms. And after some banana cells, they are transformed. We grow it to a banana tree. So for the banana cells, no matter for the banana leaves, for the stem, or even for the banana itself, they contain the cells with this GP5 gene. And then for the cells, they can produce the GP5E protein. And therefore, when we eat the banana, so we intake the E protein. And for the E protein is your self protein or the foreign protein is the foreign protein. Therefore, it will trigger our immune response to produce some antibodies, you know what I'm saying, to fight against the E protein. And then we have such immunity. And then we shall have the immunity to the and then we shall develop the immunity to this virus. After we talk about the recombinant DNA technology, the biotechnology, surely we need to talk about the potential hazard. For example, so we always think that the biohazard, the Resident Evil, they are not a dream, they are not just a game, could be a future of our human being. Therefore, the lab safety is our first priority concern for the biotechnology. And surely for the ethical issues, this time we transform the bacteria, we transform the banana. So what about next time? We transform the fish, the cats, the dog, and even human beings. And it will lead to some ethical issues.